this lesson is going to address some common issues I found students have when working with Punnett squares, and today we're just going to look at single trait crosses or monohybrid crosses. We'll leave two trait crosses for a part B to this video. It'll be a separate video. So before we do this, we're going to look at pigs. We're going to look at a trait in pigs. So we have a pig here. Pigs have two types of ears. They can have folded over ears, like this, or pigs could have, let's put the pig here, this pig has pointy ears. So you could have either type of pig. Those are two phenotypes that we'd be dealing with. We call this phenotype drooping ears. And this one we call erect ears. Stand straight up. Now, as it turns out, the pointy ears or erect ears are the dominant trait. And the droopy ears, that's the recessive trait. So we're just going to pick some symbols here that we're going to work with throughout the examples here. We will say that the erect ears, I'm going to use E for ear, we'll use a capital letter for the dominant trait, and we'll use a lowercase letter for the recessive trait. So for the examples I'm going to do here today, we're going to look at big E for erect ears and little e for drooping ears. All right, so once we set out that, let's just erase our little piggies here. And we'll just set this up. Big E is equal to erect ears, and little e is equal to drooping. Now, pretty straightforward to do a cross with a single trait, but we need to look at some language here. For example, the terms homozygous and heterozygous. Now, if we have the word homozygous, that just means that both letters that the pig possesses, both alleles that the pig possesses are the same. In other words, we could have big E, and that would be a homozygous erect ear pig, or we could have little e, that would be a homozygous drooping ear pig. The term heterozygous, and I'm sure none of this is news to anybody. The term heterozygous means that the letters are different, so that would be big E, little e. So that's pretty straightforward stuff, but where things get a little bit confusing is when you read in textbooks and read in assignments and tests the term true breeding. Well, what does true breeding mean? True breeding means that no matter what you cross this individual with, you will always get the same offspring every time. It's just another word for homozygous. So if you have a true breeding erect ear pig, that pig would be big E. If you had a true breeding drooping ear pig, which is the only way you could have a drooping ear pig, by the way, because it's recessive, that would be little e. So the word homozygous and true breeding are pretty much the same thing. So when you come across that, don't be fooled. It just means homozygous. Now, another thing that students come across is something called a test cross. That's another one that students sometimes have a little bit of difficulty with. Test cross. Now, what a test cross refers to is when you have a pig whose phenotype is known, in other words, what the pig looks like, whether it has erect ears or whether it has drooping ears, we can see, we can just look at the pig. But you can't tell its genotype just by looking at an erect ear pig. You can tell the genotype just by looking at a drooping ear pig. A drooping ear pig is going to be little e, there's no other way. But an erect ear pig could be heterozygous or could be homozygous, and we can't tell just by looking, but you can tell by doing a test cross. So let's look at an example here of, say, you've got a pig rancher, and the pig rancher, I don't know, for some reason he or she particularly likes pigs with pointy ears, so wants to have a complete herd of nothing but pigs with pointy ears. So how do you go about doing that? Well, you would breed together pigs with pointy ears. But you only want to breed pigs with pointy ears that are true breeding. You only want to breed the ones that are going to pass on a big E to the offspring each and every time. So you don't want to breed a heterozygous individual because they could pass on that little e allele, and some of the piglets you get could end up having drooping ears, and in this example, they don't want that. So in a test cross, what we do is we cross an unknown pig, an unknown, now it's unknown genotype. In other words, we do know something. We know that it's got erect ears. We wouldn't bother breeding a droopy ear pig if we didn't want any droopy ear pigs. So we've got an unknown, and we're going to cross it in a test cross always with the same thing. You always cross in a test cross an unknown with a homozygous recessive individual. Always homozygous recessive. That's what makes it a test cross. So the unknown pig, well, we do know at least one thing about this pig. We know that it must have at least one big E, or it would not have erect ears. What we don't know is what the other letter is. Now, in this particular example, we're talking about if that was another big E, well, then that pig would be saved for breeding purposes. If it's a little e, well, then you wouldn't save that pig for breeding purposes. All right, so we cross with a homozygous recessive little e, little e. Now, this is what we call our P generation, our parent generation. And what we would need to know is what happens in the offspring. We call the offspring the F1 generation. Now, for the offspring of this particular cross, let's just make up some numbers here. Let's say that there are 8 little piglets that are raised here, and of the 8, 6 are erect ears, and that would leave, well, we said there were 8, so that would leave 2 with drooping ears. All right, now, it doesn't matter how many we have with drooping ears. If we even have 1 with drooping ears, if we have 50 piglets and 1 of them have drooping ears, well, that would tell us that we must have a little e right here. Because the only way that could happen is if each parent gave the offspring, these offspring right here, must have 2 little e's. So each parent has to give a little e. This parent can't give both little e's because a parent can only give 1 letter. So to get 2 little e's, we have to have a little e in the original. So the purpose of doing a test cross is to figure out the genotype. Now, let's look at another situation here. Let's say that in the F1 generation, we didn't quite get those numbers. Let's say that we had 8 piglets and all 8 had erect ears. Now, at first glance, that might lead us to believe, well, okay, then this has to be a big E, right? But that's not necessarily the case, because with only 8 offspring, it is possible that all 8 of them received a big E from this pig and a little e from this pig, and all of them are big E, little e. Even if this was a little e, that is a possibility. It's not likely, but it could occur. So if we had 8 all with erect ears, then what we would say is that the most likely genotype for this parent is big E, but it's not the only possibility. It could be big E, little e. But if we have, say, 1 with drooping ears, that'd be little e, well, then we know it's not a big E. We can be absolutely sure this isn't a probability, this is an assurance that we have a big E, little e. So those are a couple issues that come up with single trait crosses. Another issue that I've seen come up with single trait crosses is when unusual symbols are being used for traits. Now, I would call them unusual symbols, although for geneticists they're quite normal and usual. Let's look at an example where we don't necessarily use a capital letter for a dominant allele and a recessive letter, sorry, a lowercase letter for a recessive allele. Let's look at an example with fruit flies. And let's look at fruit flies and their body color. Now, when it comes to fruit fly body color, there's a number of different variants, but let's just look at this one, where we've got something called EB positive, which is equal to gray body color, and we've got just EB without the positive, and that's equal to a black color that's called ebony body color. And as it turns out, gray body color is dominant over ebony body color, so that's recessive. So sometimes students have difficulty with this because it's not obvious. We don't have big E and little e or big R and little r. We've got other symbols. But when we write out the genotypes, we still write out the genotypes in a similar way. For example, if we had a cross with a heterozygous gray fly times an ebony fly, we would still work things out the same way. We could do a Punnett square the same way we do it with just a regular capital lowercase letter. For example, a heterozygous gray fly would just be EB positive. We put the dominant allele first, and then the recessive allele second, times ebony body fly. Well, we're not told that the ebony body fly is true breeding or homozygous or heterozygous, but to have ebony body, it would have to be homozygous because that's a recessive trait. So we just write that out as EB. Now, let's not confuse it with a two-trait cross. The EB is a single allele. It's not an allele E and an allele B. It's a single allele called EB. All right, so when we set up our Punnett square for this, we would, this is our P generation. If we want to find out our F1 generation, we need a Punnett square. So when we set up our Punnett square, we just separate the alleles just like we would do if they were a capital lowercase letter. So we have an EB positive, and we have an EB. And down here, we just have an EB and an EB. And we would fill out our Punnett square, putting the dominant allele first. So in this case, we'd have EB positive, EB. Here we would have EB, EB. Here we would have, again, the dominant allele goes first, EB positive, EB. And then lastly, EB, EB. So what we'd end up with here, in terms of genotypes, we would end up with two heterozygous to two homozygous, two heterozygous with gray body color to two homozygous ebony body color. We'd break that down to a 1-to-1 ratio. So our phenotype ratio would be 1 gray to 1 ebony, with these two being the gray and these two being the ebony, and 2 to 2 goes down into 1-to-1. All right, so sometimes the symbols get a little bit more complicated, but it doesn't change the way you do the Punnett square. It works out the same way. So let's take a look at a couple of examples. Here we've got a question involving cystic fibrosis, the most common genetic disorder among Caucasians, affecting 1 in 2,000 Caucasian children. The cystic fibrosis allele results in the production of sticky mucus in several structures, including the lungs and exocrine glands. Two parents who are unaffected by the disorder can have a child with the disorder. That last statement is key, right there, because that tells us that this cystic fibrosis is a recessive trait. Now, how do we know it's a recessive trait from this? Well, if two parents who are unaffected have a child with the disorder, well, if it were a dominant trait, if the two parents were unaffected, they would have to be homozygous recessive, which means their child would have to get recessive alleles, so the child could not have the disorder. If the child has the disorder and the parents don't, that tells us it's absolutely for sure a recessive trait. So in the question it says, these parents who are unaffected by cystic fibrosis are planning to have another child. What is the percentage probability their child will be affected by cystic fibrosis? So we just need to set up some symbols. Now we could use C for cystic fibrosis, but I don't like to use C. And the reason I don't like to use C as a symbol is I just wrote a C on the screen here. It's not a capital C or a lowercase c. Well, you can't tell. Some might say, well, that's capital. Well, no, that's capital, right? You can't tell by looking at a C. So maybe for cystic fibrosis, let's use an F for fibrosis. Let's get my pen back here. Why is my pen not working? All right. So big F, we'll say, is well, this would be non-cystic fibrosis. Or we'll just say normal. I don't like to use the word normal for that, but not having cystic fibrosis. And then little f would be cystic fibrosis. So we're using a lowercase letter. We're keeping the symbol simple here. So this is a fairly straightforward question. We have a P generation where the parents who are unaffected by the disorder have a child with the disorder. Well, if the parents are unaffected by the disorder, they must have a big F something. Could be another big F, could be a little f. That's one parent. The other parent, same thing, big F with something. We don't know what that is. Could be a big F or could be a little f. But in the F1 generation, we are told that they have a child that has cystic fibrosis. right here. They can have a child with the disorder. So they can have a child that is little f, which means the parents are big F, little f. So since we know that they can have a child with cystic fibrosis, we know that both parents are heterozygous. So that just sets up a very straightforward Punnett square for our F1 generation. We just separate the alleles, big F and little f, big F and little f. And we just fill this in. Big F would have no cystic fibrosis. Big F, no fibrosis. Big F, no fibrosis. And little f, that one would be cystic fibrosis. So we're asked for the probability that the next child will be affected by cystic fibrosis. Well, it doesn't matter which child we're talking about, first child, second child, third child. Every child is a separate instance in terms of probability. So our probability here would be 1 out of 4. And we're asked for a whole number percentage here. So a whole number percentage is 25% chance of cystic fibrosis. That's about as easy a question as you're ever going to see. The only trick to this one is making sure that we know what the genotypes of the parents are based on the fact that this is a recessive trait. Now let's take a look at another example. This one involves test cross. So it's very similar to our pig question earlier. It says farmers who raise sheep for wool try not to produce offspring with black wool. Black wool is very brittle and difficult to dye. Therefore, white wool is more desirable. If you've ever heard the expression black sheep of the family, the undesirable, well, black sheep are undesirable for the reasons given here. Difficult to dye and brittle wool. If a farmer purchases a white ram, he will generally carry out a test cross to determine whether the ram is heterozygous or homozygous for white wool. Of course, if it's homozygous for white wool, it would be breeding stock. If not, well, then it wouldn't be. So white wool, big W, is dominant to black wool, little w. Now, I don't like using W's because you can't tell a capital from a lowercase, but those are the letters we're given and might as well stick with it. So, in the question, it says, if the ram is heterozygous for white wool, so a ram is heterozygous for white wool, meaning big W, little w, the expected phenotype of the offspring of the farmer's test cross would be. Well, we need to know what the ram's being crossed with. Of course, the ram's being crossed with the ewe, female sheep. But what is the genotype of the ewe? Well, the key to this question is that it tells us that it's a test cross. And the test cross, we always cross with homozygous recessive. So that's little w, little w. So all we need to do here is set up a Punnett square. That's our P generation. So in our F1 generation, we separate the alleles of the parents, big W, little w, and little w, little w. And what we end up with, big W, little w, little w, little w, big W, little w, and little w, little w. So what we would expect here is half of them being black, sorry, half of them being white, because white is big W, and half of them being black. So our correct answer here is half black, half white.